All right, so let's talk about chapter 2, the tools of the laboratory. This chapter actually will be very beneficial both for um, the lecture component of the class as well as the lab component. So I will actually be uploading this into both the lab shells and the lecture shells. So chapter 2, tools of the laboratory. In microbiology, we talk about the 5 I's. We talk about inoculation, we talk about incubation, we talk about isolation, we talk about inspection, and then finally identification. And we'll talk about each one of these in a little bit more detail. Inoculation is the concept of growing bacteria. The culture is the bacteria that we're growing in the lab. The stuff we grow the bacteria on is referred to as a medium. The plural for that term is media. These are nutrients found that help bacteria grow. A small sample of the microbes that are placed onto the growth medium is referred to as the inoculum. The process of putting that inoculum onto the medium is referred to as an inoculation. And this is how we can collect clinical specimens from body fluids, from body discharges, from certain anatomical sites, nose, ears, eyes, mouth, etc., or diseased tissue. So any of these things can be a place where you collect the inoculum and then inoculate that inoculum onto a media in hopes to grow a culture. Incubation is done in an incubator. Incubators can be very simple structures. The ones we have on our campus are about half the size of a refrigerator or so, but they are temperature controlled. These are used to grow bacteria. They can be set at a variety of temperatures. Most labs you will go into will have several incubators set at different temperatures because certain microbes grow better at some temperatures while they don't grow so well at others. So you would have an incubator set at, say, 25 degrees, and then one set at about 37 degrees, and then another one set about 45 degrees or so. Sometimes atmospheric gases such as carbon dioxide or oxygen may be required, and we can, in certain types of incubators, we can account for that and make sure that we are getting those proper levels of gases. And sometimes during the incubation period, the microbe multiplies and produces growth that is observed macroscopically. That's what you see when you put a petri dish into the incubator and you don't see anything. And then 36 to 48 hours later, you open it up and, oh my goodness, there's all this stuff growing on plate. Here's the various types of cultures that we'll use in the lab. This is a pure culture here. Each one of these tubes contains a single bacteria type. This one maybe is a staph organism of some sort. This maybe is a strep, and this is maybe some other bacillus organism. Mixed cultures are where you can see, if you look closely at this culture here, you see some yellow dots and some white dots. Well, the yellow dots and white dots are being produced. Those are called colonies, and they are being produced by different bacteria. Therefore, you have a mixed culture. And then you have over here what is called a contaminated culture. In our labs that we do here on this campus, probably the most common thing that we see that we really don't want to see is a contaminated culture. The red, whatever this red stuff is here, is what we're attempting to grow. But we've got this big glop of white stuff here. That's contamination inside of our culture. Different types of medias that we will work with in lab. The physical state, be it a solid or a liquid. Chemical composition, what does it have in it? Does it have salts? Does it have sugars? Does it have proteins? And finally, the functional type, what is the purpose? Is it there to show us a pH change? Is it there to show us a gas production? Is it there to demonstrate something along those lines? Again, the 3 physical states of media are liquids, semi-solids, and solids. There are 2 different types of solids, those that can be converted into a liquid and then those that cannot be converted into a liquid. For the most part, in the type of labs we do here, we want this type of solid media. If we're going to use solid media, we want one that cannot be liquefied. Although there is one particular type of solid media that can be liquefied. We'll talk about that one in the lab. A semi-solid is usually going to be in found in a test tube. And if you think about the consistency of, say, Jello, so a semi-solid is usually like a sort of a soft Jello. This word right here is another one of those words that over the years has changed in pronunciation. I was taught, again, many more years ago than I care to think about, that word was pronounced auger. Now most people pronounce it agar. I don't, you say tomato, I say tomato. It's the same product. It is made up of a complex polysaccharide isolated from a genus of seaweed called Gelidium. It is solid at room temperature. The advantage of this product is that it does not liquefy till it gets to almost 100 degrees Celsius. And you can ask Siri to convert 100 degrees Celsius to Fahrenheit temperatures, and you'll find that 100 degrees Celsius is somewhere in the neighborhood of 180, 190 degrees Celsius. So you see that's quite warm. However, it doesn't re-solidify until 42 degrees Celsius. So once you've melted it at 100 degrees Celsius, then you've got a long time at room temperature before it re-solidifies. So you've got a long time to work with it. If you're needing to pour it into tubes or pour it into plates, after you boil it to make sure that it's sterile and you've liquefied it completely, then you've got a long time to get those plates and tubes poured. Agar is flexible and moldable, and you can put it into tubes. You can make what are called slant tubes, and we'll talk about those in a few minutes. You can put it in plates. You can put it in all sorts of things. And the real benefit it has for our microbiological studies is that it's not a digestible nutrient for most microorganisms. Most microorganisms, if you just take a plate that has nothing but agar in it, most microorganisms won't grow. That way, when we put glucose, for instance, in an agar, and the bacteria suddenly start to grow, then we know those bacteria are metabolizing the glucose and not the agar. Again, here are some different types of tubes. There's the liquids, there's the semi-solids again, and the solids, which are can be reversible to liquids. Chemical content of media, the defined or synthetic media. A defined media is one where the composition is precisely chemically defined. We know exactly what's in there. These are going to contain a pure organic or inorganic compounds that vary little from one source to another. Basically, what that's saying is if you get a particular type of agar from company A and you get that same type of agar from company B, they should not be different. They should contain the same things and in the same amounts. They should not be different. A complex media, on the other hand, is one where the components are not chemically defined. In other words, they may contain extracts from animals, plants, or yeasts. In other words, they just have a protein source. Maybe it's an animal protein, maybe it's a plant protein, or maybe it's some sort of a fungal protein, but it's still a protein source. Blood, serum, meat extracts, milk, yeast, soybeans, peptones. Peptone is a protein source. All of these things can be added to medias to make what is called a complex media. If you wanted to grow, for instance, Staphylococcus aureus, more commonly referred to as Staph A. If you're going into the medical field, you're going to learn a lot about Staph aureus. The chemically defined media that is particular for growing this organism will contain 0.25 grams each of these particular amino acids. Don't worry, I will never ask you to remember these things. This is simply an example. It will contain half a gram each of these amino acids. It will contain 12 1/100 grams of each of these amino acids. And then there are additional ingredients, vitamins, thiamine, nicotinamide, pyridoxine, and micrograms of biotin. There will be certain salts. All of these ingredients will be dissolved into 1,000 milliliters of distilled water. Then using a buffer compound, which, you know, if you've taken chemistry, you know what buffer compounds are. Those are things that modify the pH without altering the product that you're making. So you add 1,000 milliliters and then you buffer it to a pH of 7, which again, that is a neutral pH. And then you have then made 1 liter of medium for growing Staphylococcus aureus. Now, this would be a liquid medium because all it contains is water. If, however, you wanted to grow these on a solid surface, you would then add an appropriate amount of agar powder to this 1,000 milliliters of distilled water, and you would then make what is referred to as a nutrient agar. Brain heart infusion, another item that you can use for growing Staph aureus. Again, not going to ask you to remember all of these numbers and grams and things. Just another example of a complex medium made for growing a very particular type of Staph aureus. A general purpose media is one that is used to grow a broad spectrum of microbes as possible. These are generally complex medias. They're generally going to contain a lot of different growth nutrients, so lots of different things will grow on them. Mostly what we're going to use in our lab on this campus will be a general purpose media. We want as much to grow on there as we can. An enrichment media is one that contains complex organic substances such as blood, serum, hemoglobin, or some special growth factor for the growth of microbes which we refer to as fastidious. A microbe that is fastidious is one that requires some very particular growth factors. There has to be a blood product there, such as blood or serum or hemoglobin. Maybe it requires a very particular temperature range. Maybe it requires a very particular pH range. All of those things would make that particular organism fastidious. There are some examples of different types of media. That is a blood agar. That is an organism growing on the blood agar. And this area around where the agar has changed color is an example of something called hemolysis, which we will talk about later on in the semester, that summer, and later on in the lab. A selective media is going to be a media that contains certain nutritional agents that are going to inhibit the growth of certain microbes or microbes. But in addition to inhibiting the growth of some, it's also going to promote the growth of others. These are very important in primary isolation of specific types of microorganisms. If you have an unknown sample, you have a selective media that will only allow gram-positive things to grow. You spread this unknown media on there, or unknown culture on the media, and it starts to grow. Well, you know one thing about your unknown culture, at least it's a gram-positive organism. These help speed up isolation by suppressing unwanted background organisms and favoring the growth of the desired ones. Again, you know you have a pure culture, you just don't know what it is. You put it on a medium that only grows gram-negative things, and it grows, then it's a gram-negative. If it doesn't grow, then chances are you've got a gram-positive item working there. Differential medias are a type of media that are going to allow lots of different things to grow, but they're going to display a difference in how they grow. Maybe there'll be a variation in colony size or color. Different organisms may color the media different. Some organisms will be pink, while other organisms are maybe yellow. Gas bubbles may be produced by a particular organism. And these variations will often come from chemicals in the media with which the microbes react. So a particular chemical reacts a very particular way with a certain microbe producing in a particular color. Then you can then extrapolate that your organism is this because of that particular color change. And then here's an example of selective and differential medias. Here's my mixed sample in this tube. We put it onto a general purpose media and we see varying different sizes and colors of organisms here. But over here, only one species is growing. Yes, they're all different colors, they're different sizes, but they're all the same color. Whereas over here, it's kind of hard to distinguish, but these ones and these ones are different colors. These are darker than these. Over here, you have, again, a mixed sample growing on the general purpose media. So you see all these different things growing. On the differential media, you see all three of them are growing. But over here, you see very different colonies. You see some white colonies, some sort of bluish ones, and sort of red ones. And if you know what this media is, and you know what types of organisms grow to look like that, and what looks like that, and what looks like that, you now know what's in your mixed sample. Selective and differential medias can be both. A medium can be both selective and differential. An example that we will sometimes use in our lab is called McConkey's agar. It suppresses the growth of some organisms while producing visually distinct colonies between ones that do grow. That's because the dyes used as differential agents have different pH indicators. That changes the color in response to the production of an acid or a base. So here's something growing on McConkey's agar. You've got this structure growing here, and these growing, and then you see the different colors forming on the different organisms. Finally, some miscellaneous media. We have a reducing media. We don't use reducing medias in our lab, and we don't use transport medias, but it is important that you know what these things are. Reducing media is one that contains a substance that absorbs oxygen or slows the penetration of oxygen. These are important in growing anaerobic bacteria. One reducing structure that we use many times is something called activated charcoal. Activated charcoal under certain conditions will remove oxygen from the air. If you put it in a chamber, you put some activated charcoal in that chamber, then you put a few drops of water on it, seal that chamber up. The reaction of the water and the activated charcoal will draw the oxygen out of the air. A transport media is one that is used to maintain and preserve a specimen that has to be held for a period of time before chemical analysis. If you've ever been to the doctor, for instance, and they've taken a blood sample, and you'll notice in the bottom of that blood tube, there's a little stuff that looks kind of like wax or gel or something in the bottom of the little tube. That's a transport media. That's in there to keep the blood from going bad while it's being transported from point A to point B. There's our some other miscellaneous medias, carbohydrates media. These are different types of media growing. All of these 4 tubes started looking like this. This is the control, so this is what all 4 tubes look like at the beginning. If the bacteria, as it metabolizes whatever this carbohydrate is, if it raises the pH and makes the pH go up, you see the product gets a darker red color, get this sort of purple looking color where you can see it there. If it drives the pH down below 7, you will see it turn yellow. This is for acid production without gas. However, if you get an acid production with gas, in addition to the tube turning yellow, you see that there is a visible bubble now forming in the tube. This little upside down tube here is referred to as a Durham tube. Durham, just like the city in North Carolina. And its function is to trap the gas that is being produced by the bacteria that are growing down there. An assay media is one that is used to test the effectiveness of antimicrobial drugs. We will do a little test toward the end of the semester using antibiotics, and we will perform sort of a little assay. Again, these types of media are used by drug manufacturers to test the effectiveness of things like disinfectants and antiseptics, to look and see if their cosmetics are clean enough to be sent out into the community, and to check on preservatives and microorganisms. An enumeration media is one used by industry to count the number of organisms in things like milk and bottled water and food products and soil and other samples like that. All right, I'm going to take a break for a few minutes, let my throat rest, and we will come back and continue with Chapter 2, starting with isolation.